So our second talk is by Ashlee Ford, and she's going to be talking about multiscale simulations of lung fibrosis. And I will turn it over to her without more ado, and I will make a 5-minute warning if that's okay.
Excellent. Okay, let's get this shared here. All right, seeing the PowerPoint now. Okay, so I am excited to be here to present to you all today, and this work is highly collaborative. We're building a multi-scale lung tissue simulator and looking at the infection of SARS-CoV2 and specifically tissue damage. And so a lot of what we're doing is going to focus on the lung epithelium and incorporating different scales of effects. So I am an associate professor in the Department of Chemical and Biological Engineering at the University at Buffalo, the State University of New York. The most important person for today's presentation is Mohammed Amin-ul-Islam, who's the postdoc who has put an extraordinary amount of effort in the last year on this project. So he, I think, is on the call today, so I would invite him to join us for the question session today too. So there's a larger collaborative team, and I know that you all have, or this IMAG MSM group has heard from other components of this before. So Paul Macklin, James's colleague, has organized this collaborative group. And then the two people that I've worked most closely with outside of my team, Thomas Hillen and Amber Smith. And if you're interested in the larger project, I encourage you to go to our PhysiCell page that has open access versions of all the codes and things that you can join in on and play with and explore and add to and contribute. So we are very interested in modeling the physiological processes that happen due to SARS-CoV-2 infection and the immune responses, and then thinking also about downstream types of consequences. And so I'll show you different kinds of images like this about different processes we're interested in, about things that get disrupted due to the infection and the overall immune response. So we're considering COVID-19 as a multi-scale problem, and we do in the model consider subcellular effects where the virus binds to the ACE2 receptors and moves into healthy cells, and then where the virus itself can replicate and can release viral particles. At the cellular level, we have different agent-based rules for the death rate and the secretion of various other signals that can communicate between different cell types. And then we have the immune response where immune cells can phagocytose dead cells, attack infected cells. The tissue level we have where the viral released virions can move through the tissue, the infection can spread, the tissue damage can spread, and both the immune cells and fibroblast cells are coordinated with various secreted factors. There's also a level of the model that communicates out to the lymphatic system to recruit more immune cells that then could come in through the vasculature and respond. And my team is also interested in other things like the change in systemic angiotensin 2 in response to this, but I won't show you anything about that today. So the most recent version of our preprint has this kind of showing some of the immune dynamics and the intracellular viral part. So given a healthy epithelial cell in the lung, so thinking about this lining the airways or lining the alveolar region, that the virus can come in, represented by this orange dot. There's some different viral replication processes that happen internally inside of each epithelial tissue agent that then secrete various factors. There's a whole cascade of immune cells, both innate and adaptive immune system, that respond. And I won't go into those details, but you can ask about them if you're interested later. But there's a whole sub-team on the project that focuses on this immune response, and I was not part of that group. We also have this lymph node part The part that's more interesting to me is when the T cell response comes in, and T cells can detect different inflammatory signals and the infection in these cells, and CD8 plus T cells can induce the death of these infected cells. We, on my team, were interested in how does fibrosis happen in response to this CD8 plus induced T cell killing and this latter stage in acute respiratory distress when fibrosis starts to emerge. So it does not happen as part of the initial immune response. Data from Amber Smith's lab had shown that the fibrotic and more of the tissue structural changes were happening after the peak in T cell response. And so that's why in a rule-based fashion, we were considering this CD8 plus T cell response to be the main sequential trigger for our rules. So we considered this adding fibroblasts, and that as soon as this infected cell is killed, then there would be an anti-inflammatory cytokine signal that would start. We considered two phenotypes of fibroblasts. So we'd have an inactive phenotype that's in the host tissue, so already residential fibroblasts in the lung tissue, and then once they sense the anti-inflammatory, they become activated, but then there's also recruitment. So the active phenotype starts to secrete collagen, but that inactive does not. We have this, I've already talked about how this anti-inflammatory cytokine being triggered, and right now we have a very simple rule that is when that happens, that at that location becomes a constant source of a secreted anti-inflammatory, more generic anti-inflammatory molecule. We're considering it as basically would be a TGF-beta response, and that is what the fibroblasts that are resident and that are recruited are going to hone towards. So fibroblasts are going to move up the gradient of this anti-inflammatory cytokine and start to secrete collagen along that pathway. So this is just a representative simulation of our agent-based model. It's a hybrid model. There's some PDEs in the background for how the virions are moving around, how the secreted factors are moving through this tissue domain. So the epithelial cells are this light blue color or this medium blue. They don't move around. There's a number of immune cells that are moving around. You see the neutrophils are moving quite rapidly as a response. You'll start to see the T cells coming in, and they'll be more aggressive as well. The macrophages were part of the innate immune system. That's why they were there so early. And so you start to see that cells get infected and are grayed out. And if they are killed, we leave behind a little black dot for a little bit, and then it becomes white when that space has been cleared out or the debris from the dead cells. We are now simulating the fibroblasts as these purple spheres in this particular movie. And so we'll see that the fibroblasts are honing towards the gray regions to respond to where there has been cell death, to try to replace that missing epithelial with collagen fibers. So physiologically, what we expect to happen is that after tissue damage, that the body would try to repair that by building collagen fiber networks and healing the wound. But in some cases, it can get too tilted towards fibrosis, and you may end up with a substantial scarring, and we want to be able to simulate when there's too much fibrosis and then when there's not enough as well. So that's represented here. So we can do those kind of simulations. The color scheme was slightly different in this earlier schematic here with these immune responses I'm showing, but we can simulate different conditions where we have different of the immune factors turned on and off, the rates at which they do various processes. And so in all of these, we had substantial tissue damage, being that there was this white region that was not acellular that would develop, and substantial immune infiltration when we turned on the immune response. My team sought to add the fibrotic aspect to this, that we would fill in these blank spaces with this rule-based fibroblast response. So In this simulation, the fibroblasts are represented as the small orange spheres. And so this is showing the collagen field evolution that would arise in those blank spaces based on after the infected cell was killed. There's this anti-inflammatory signal, fibroblasts hone to that location and start to create secrete collagen. And then the collagen is not mobile, does not diffuse in this simulation, but it gets secreted where there's a stronger signal of anti-inflammatory cytokine and when there are more fibroblasts that had honed to that region. So we're starting to see significant regions filled in with this collagen field. So Aminul added that portion, and then he simulated this under various conditions where we had a moderate disease condition of, so we can control in the simulation environment the multiplicity of infection that is related to the initial amount and distribution of the virus exposure on this tissue segment, and by changing the time required for the CD8 plus cells before they kill an infected cell. So he chose the settings that would give us this quote unquote moderate condition, and we could see then the evolution of the collagen in response to that. He also tracked the emergent number of fibroblast cells, CD8 plus T cells that would respond to that. And we have some collagen secretion before we recruit substantial amounts of fibroblasts. So basically our rules say that if it's inactive, it is at a steady state. It's not there's not more fibroblasts. Fibroblasts aren't able to leave. So it's a flat line. But as soon as we start to get that signal, the fibroblasts can start to move. And we also then turn on their birth and death rate at that point. So the model has where they can move away and can die out, but then there's this massive recruitment that happens after some lag time. So we start to see that, but then we get substantial collagen once these recruited fibroblasts move into the zone. We also did the more severe conditions. So you'll note that by the same 10-day time point, there's far less of the resident tissue, the host epithelial tissue that remains, and you have much more substantial collagen that has formed, and you have quite the infiltration of the immune cells here in this tissue. So again, just by changing the multiplicity infection, you got this different severity case, and can see the different collagen and fibroblast T cell responses. There's many other things that this model will give us. We were just interested in more about these questions about the collagen aspects from my team. So we are working on considering the ways that this collagen field might affect the diffusivity and the transport rates of different cells and the viral particles. We're also interested in the cellular regeneration aspects of the tissue, not just this fibrotic part. We're also considering how collagen deposition can exert forces in the surrounding area and those conditions that would lead to it being pathologically pro-fibrotic in a way that is unhealthy. We're also considering if we should be looking at collagen alignment and porosity effects and whether that's relevant at the scales that we're interested in as a larger team. And then there's several other aspects that are being incorporated by the entire tissue simulation coalition on refinements to the immune response part, refinements to the viral replication dynamics, and incorporating new data as it emerges specific to SARS-CoV2 rather than more infectious disease generic types of numbers that may be in there right now. Everything can be is in as a parameter and nothing's hard coded, but sometimes our representative parameters are inspired by influenza infection or the original SARS infection, places where there were established data. So, so that I think is the end of my presentation. I want to definitely again though acknowledge Aminul and his work on this project.
Okay, thank you very much. That was a very, both of our speakers stayed to time, which is an unusual virtue, so I appreciate it. that means we have time for some discussion. So please take advantage of that. And then again, if we can try to start with less technical questions and ideally from some of our, not our usual suspects and questions, then we can always go and have more technical questions afterwards. I'm sure there are a lot of interesting issues for both speakers. Some weeks we have two talks that are very parallel to each other. Here we had talks that were very orthogonal. One that was beautiful mathematics about how to build models and how to analyze models, and one very practical thinking about how to translate biological observation into meaningful multi-scale models. And so that contrast is one of the serendipitous kinds of things that happens in this meeting, which I really appreciate. Thank you to both of our speakers. And if people have questions, please feel free. You don't have to raise your hands, although if you want to raise your hand, I'll try to keep an eye out and see who has requests.
So I wanted to ask something from Ashlee. So if A, thank you, this was fun, and B, if you tried to coarse grain this, would you try to do something in terms of averages, in terms of densities, or would you also put some spatial structure? What would be the coarse grain model that would make you happy? In terms of what variables, not what operators?
Meaning that you would want to, if I was to coarse grain this to get to like whole lung type of fibrotic processes,
no.
or
if you look at the simulation that you showed us, of course one can say what is the average number of so much and so, but do you think that in terms of reporting, would you want to only keep the average density of each of a few species, or would you also like to have some sort of correlations, like pair correlations, some measure of the microstructure of it? Do I make sense in my question?
I think so. So I know I'm trying to think about how to articulate an answer to this. We know that there would not need to be, or having information like the average density of collagen would not do justice to this, okay, right? So we would want to be able to think about if it was something, a simple thing to extract could be the peak collagen, or if it's sort of as dense as it can be, has it reached like a maximum or a close to, impenetrable or very dense region where that's very fibrotic, and that the rest is sort of okay fibrotic. And so that would be a piece that we would want to know some kind of resolution to. For the other aspects, I think on the immune side, there's knowing whether things are sort of concentrated because they're actively moving there or just because they're near the boundaries. So if there was something that could tease apart sort of the activity level of the immune response, that's not just cell population level, but sort of like how numbers of interactions or something like that. That also could be something that could be determined from like a network kind of analysis. So, and still relevant to these types of problems.
Okay.
I want to second Yannis's comment, which is that these spatial multi-scale models, which of course I work on as well, especially stochastic ones, they produce these beautiful movies. And yet when we do analysis, we tend to do averages, maximal values, areas under the curve, at most first order neighbor correlations. And all of those lose a lot of the complexity of the spatial structure. Mm-hmm. And yet when we try to look for the more sophisticated spatial metrics like topological homology metrics, you have to have very, very large simulations. You need very, very large data sets to be able to do anything with them, bigger than you have available in experiment. And so one of the problems that we face is that we really don't know how to compare stochastic images or stochastic time series. To ask the question that Yannis asks all the time, which is, do these belong to the same universality class or not? Were they generated by the same mechanisms? This happens if we look at two sets of blood vessels. And we see a vascular tree, and we look at the vascular tree, we might characterize it by the branching angle and the interbranch distance. But in fact, you could have vascular trees that have the same branch angle distribution, the same interbranch distance, and have totally different structures. And so there really is a question, which is that there's so much information that our eyes see and appreciate in these kinds of models, the ones that Ashlee showed that are so beautiful. And yet when we do metrics on them, we throw out almost all of that spatial complexity. And so the insights into the mathematics from the mathematicians about how we could actually use and respect that spatial complexity, I think it's a really hard problem. So ideas about how to do that would be very valuable.
Yeah, and you know, in the time to, you know, we showed one movie, right? Each time you do these stochastic simulations, you get different movies, and we often will average or show a representative one, but we are interested in things that might be rare events that can also emerge, and trying to detect those and to see that they're not just part of the, you know, is it, if we run this 1,000 times, we'll get mostly the same things, or do we ever get anything that's different types of phenotypes that might emerge, and how to like characterize those. And so there's lots of interesting future challenges with analyzing these systems. Thanks, James.
I have a question for Ashlee. Hi, Ashlee, I'm Greg.
Hi.
We work a lot with folks in the Lung Institute here that have clinical practices, and we do a lot of autopsies on people who have died of COVID. Are you using, what kind of data are you using in order to, I mean, I can imagine I do things similar to what you're doing here, and I can imagine there's I don't know how many parameters, but there's a lot of parameters in your model. And you showed hints of what sensitivities exist if I start playing with those parameters. What kind of truth data do you have in terms of clinical data or autopsy data? We have, for example, now probably a couple dozen lungs that have been autopsied from people that have died of COVID. And you're making predictions about certain locations in the lung. And my colleagues, who would never let me in doing the autopsy, but they tell me the results, and the results are something that I think you are making predictions of, and that you could potentially use as truth data for the things that you're doing, and maybe learn some of the parameters for which you only have good guesses for.
Absolutely. So this team got started April of last year, and sort of the main, I'd say our biggest momentum was from April until November. Now we're in like the polishing stages and going back to see now what is published, but the idea to build some infrastructure based on physiological mechanisms and processes that could be involved, but knowing that the actual magnitude of certain aspects were not yet known or quantified in the context of COVID-19, and to be ready to be able to put those in when those became available. So there are, some of the things are actually parameterized to data or have diffusion coefficients with reasonable magnitudes, that kind of thing. But some things are, you can actually put in any diffusion coefficient or any, but they're left as, here's the thing you would need to specify, you could put them in, you can run the simulation. We also, some of the thing, or a lot of the species are normalized where they're set from 0 to 1. And so you could then say, well, I know that this one in the future will want to weight those slightly differently or very differently. So we definitely are at the stage now where there's things that are emerging, like the data you talked about, that I think the first product from this group will be this framework, and then the sub teams are working on doing that validation to data so that we could have, for this particular case, I'm going to run it with actually realistic things. I'm going to do parameter estimation, and I'm going to do a direct validation. And I can take this piece and now use it. But then other people could use it for other types of infectious disease responses, but have this framework that exists that then they can customize and pull out elements of, or use the whole thing, or run many of these kind of plaques in parallel and examine larger tissues, things like that. So, so that has definitely not been completely done yet on this team, but it's definitely an interest in the future. And if people have that kind of data and want to partner, I think there's lots of opportunities. And the first author on this, I noticed Michael is on here. You know, we're very interested in those aspects. So thank you, Greg.
And I'm asking because related to what Giannis and James both said is that when we get clinical data, it's very coarse-grained and it's static. And so we have snapshots of very advanced disease, and we are asking, does anything we do replicate? You know, so you're doing a really coarse-grained learning mechanism, right? Do I get disease structures? I mean, my colleagues who do these autopsies say the damage, first of all, they don't know what got healed. That's a real problem, right? You only know what's still damaged, and you don't know. But given what they do see, what they observe is incredibly patchy disease. It is not a continuous infection of the lung, and they have very specific areas of the lung in which they can go in and tell us coarse-grained information about, you know, densities of, you know, the patches of infected cells, how many have died. They observe cells that burst. When they do die, they dump all their contents. So there's all kinds of, I mean, I am empathizing with you because in the work that we do, we're really struggling to ask what kind of data can we get that would allow us to go back in and do sophisticated learning of the things that we don't know. Anyway, very, very impressive, you know, amount of work, a ton of work. I can't imagine how many man and woman hours are involved in this, but it looks pretty impressive.
So other questions?
I have a question for Giannis.
Yes.
The tools that your team is developing, are there any software distributions that you have or others where like people could download your software and could apply it to their systems, or should people directly work with you if they want to try to apply some of these algorithms? Or
no, fair. As you know, in kind of recent years, everything is somewhere on GitHub.
Yep.
So you can download something and run the examples in our papers. For the other ones, I mean, okay, if it doesn't immediately go, you know, within reason, we would be very happy to either work with somebody or to just help a little bit. I mean, we don't have to kind of, how to say, enforce ourselves on papers. So we are happy to help within reason. But there is GitHub repositories for everything that is in our papers. And we are happy to maybe explain a little bit. Also, if it's interesting, we could work with somebody. But no, we do not have, as you, I'm sure, understand, we don't have the manpower, even though we would wish, to kind of make nice tools. So yes, things are reproducible, but it takes a little extra twist every time. As you know, there is no, I mean, nobody gives you money to make good tools, or at least not in what we do. So, sorry, but we are happy to help without enforcing ourselves as collaborators.
And I think Greg brought up an interesting point, which is when you have static information, you can't always, from a population of static images, infer dynamics. So my classic example of this is if you have tumor size versus time, you know the net rate of growth of the tumor on average in multiple individuals, but you don't know whether that growth rate was happening because there was slow growth and no death, or whether you had very rapid growth and a lot of death and they almost balanced. And the same kind of thing happens if you look at patches of lung damage later on. You don't know if you had many patches that were initially seeded and that grew at different rates, or if the patches started at different times. And you don't know if some of the patches grew very rapidly and other patches grew slowly, or they all grew at the same speed.
Yeah, I can say something to this. It's not a wonderful thing, but it's a useful thing. If you remember, when I said I reconstructed the space, I reconstructed something that was one-to-one with the space, not necessarily the space. The same technology can be used to put things in order, even though you don't know if it was slower or faster between. So it is still useful. You have a progression. You understand? As a matter of fact, at some point, there was a company in California in which if you gave them a whole bag of photographs that you find in your grandmother's, the box somewhere, they would put it in temporal order for you. And it is quite easy to do. DARPA had a shredder context in which they would shred the images and you could put them in order. So it is quite usual and not difficult to still use, I'm telling you the same thing. You may not have explicit time, but you can have order in time, which is also useful.
Okay, well, I violated two of the rules that I said at the beginning. I asked technical questions and I spoke a lot. So I appreciate people's patience. We'll have time for more questions for both of our speakers in a minute. I just want to say again that we would very much like the subgroup leads, you know who they are, to begin to prepare presentations for these Thursday meetings. Please let us know when you're willing to present. I think our seminars are really good, and any help you can provide in getting the word out so that we have a bigger audience would be much appreciated. Please do help us get the word out. And also agenda items for the steering group would be very helpful. There have been, as we mentioned before, some suggestions for more explicit statements of goals, and what people think those should be for this working group would be appreciated. And so we all would be very grateful. Many of the subgroups have been extremely active. Some important topics are not so active, but this is a committee of the willing, and we appreciate everyone's volunteer time for these things. So if are there any other specific business that we need to take up before we open up the breakout rooms and let people talk in more detail to our speakers? And by the way, if your breakout room becomes empty, our speakers are free to go. Of course, this is all voluntary. I hope people will stay around and have some conversation. Is there anything else that we need to have as a general talk before we have the breakouts? If not, I will, here, give me 1 second here. I will open the breakout rooms.
This is Reed Shabin from NIAID. I just had a quick question. There's a couple of funding opportunity announcements that hit the street just yesterday. Is the best place to put that information in the Slack channel?
That would be good if you would email them to me. We'll also distribute them by email to our membership.
Okay, thanks.
Thank you. Believe me, everyone is hungry, so they'll be very glad to find out.
All right, sounds good.
Thank you. Okay, Giannis, I will put, if it's okay, I'll put you in a breakout room. Ashlee, I will do the same. And again, if people don't show up, that's
we can go. Yes.